Welcome to Rocket Biologist, the show where we meet experts in medicine and biology who are helping humanity thrive in space and prosper here on Earth. I'm your host, Dr. Adeel NASIR. Welcome to Rocket Biologist. Today we are going to talk to a special guest who is expert in plant space biology. When you think about plants, they are green, they are beautiful, they give us hope, they give us feeling of connection that we enjoy going into the forest, going into a field. The product of plants can be expression of love. For example, on Valentine's Day, we destroy so many roses. They were not basically made for us, but we use them. And similarly, we eat them. The product we call fruits. They are sweet, they are wonderful in taste. All these pleasures that we enjoy on Earth, we never think about it, that if one day we are going to live in deep space, we have all those luxuries. And the answer is no. Seeing a plant on the International Space Station is just a joy for an astronaut. And our guest today is that expert who is trying to achieve that extraordinary task to grow plants in space and beyond space on other planets, Moon and Mars. I welcome Dr. Borja Barbero Barcenilla. Welcome to the show. Good to see you and good to have you on the show. How are you doing, Doctor?
Good. Thank you very much for having me. It is a pleasure to be talking about plants in space today with you.
Great. So we will start with this. I think this is a very redundant question, but I always like to ask my guests, who is Borja Barbero Barcenilla? And tell me if I pronounce your name correctly.
Very, very good pronunciation. After being in the US for 15 years, I've heard it all at this point, But yes, I am... I've been defined as a force of nature by many friends, a person that has like a very strong drive to make things happen. Now I am a father of a beautiful daughter that was born here in Texas, in the United States. I'm a person that loves sports, loves science, and loves hanging out with my friends. And I imagine we'll go into the science later. But I'm a happy person that enjoys my work quite a bit.
Wonderful. So when you hang out with friends, I'm kind of assuming that you are a foodie. You love food. So what's your favorite food?
Oh, like being from Spain, obviously, we were discussing right now, like Spanish tortilla and Spanish ham first come to my mind. But now that, I'm become Texan as well, I could go for some good Texas barbecue, some Tex-Mex, they're growing in my heart. I don't think yet to the level of Spanish ham and my mom's tortilla, but it's getting there.
Well, no one can cook like exactly. That's for sure. So are you a good cook, by the way?
No, I'm not. I can make some things happen. My wife, she's an excellent chef, She's the one that makes the very special meals at home. And, if she has to do something else, I'll put something together, but it's nothing super, super fancy. It is, it's decent, it's edible, but it's not, nothing super special. My wife and my mom, they are the best chefs ever. And my grandma.
So good you didn't, you are still in field of science, then it's better you grow plants. Yeah. Okay. So please tell us about your metamorphosis from Borja to Dr. Barcenilla.
So my path to get where I am today, it was very strange. I actually came to the US with a soccer scholarship to play college soccer. I played first at Iona College, a small D1 near New York. And then I transferred to University of Delaware. And during this time, I was a full-time student athlete, right? And I was my major was biochemistry, which is not very common for student athletes to have a major that is challenging. And while I was a student athlete, I was still doing undergraduate research where you go to the lab and you participate in different projects at the same time as, waking up at 6 a.m. to go and work out with the team, then trainings, then travel for games. So, but I always say those 4 years of student athlete life combined with research, they were amazing. And I was very thankful that I was surrounded by fantastic people as well. And then I tried to make it to professional soccer, didn't really get a solid offer that was, that excited me. So I started my PhD at Texas A&M University, and I was very happy to join the lab of Dr. Dorothy Shippen, where I was on that great lab as a grad student, and then I stayed as a postdoc for a bit while we were working our NASA project. And then very recently, a year ago, I made the jump to industry, and I moved here to Dallas, Texas. So that is a tiny bit of the metamorphosis from, how I was a soccer player that, played in Real Valladolid for all of my childhood until I came to the US, got a soccer scholarship, and eventually became a doctor in the US. A tiny bit of a windy road to get to where I am today.
Well, that seems like that's a wonderful, path you took, that from sports to science. That's amazing, because I think if you were close even to get the, qualified for a professional team, so you must have been playing really good.
I think I was, I was good enough to be where I was. I don't think I was good enough for the last step. I got some offers that were not super exciting, so didn't take them. And then I was lucky to play in USL PDL, which is like minor league, so to speak, for 3 years. And then I joined, after that, even at the grad school level, I joined the Texas A&M men's club soccer, where I played and then coach. And, I still follow my passion of soccer. I coach local teams here in Dallas. I coach a team called Station. Soccer is part of my life, of my culture. Me and my wife, we enjoy watching games. The team that I support in Spain sadly is very bad right now. We're in second division, and I still have some colleagues playing on the team, which is great. I suffer way too much due to soccer sometimes.
Oh, wow. So you're this aggressive soccer fan, who fight after every match?
No, never fight, Okay. My team loses all the time, so there's really not much of a fight.
Okay, wonderful. So how did you become interested in the field of space biology?
I think, as almost everyone that gets into space biology or space science, we are all motivated by sci-fi movies, right? So growing up, I'm a huge Star Wars fan, right? I've seen all the movies, all the shows, big Star Wars fan. Growing in Spain, the sad reality is it's almost impossible to become part of a space biology project, right? Just because Spain is not, it's not strong on that space, right? Never said. But once I was in the United States, I joined a lab that was working on plant telomeres, and we were the pioneer lab working on that project. And it was a very random message that we got. We got an email from an Argentinian astrophysicist, and that he was very interested in studying telomeres in plants in space. And this was all motivated by the twin study experiment, right? In which one twin remained here on Earth, and the other twin went to space. And after 1 year, one of the major changes that they saw was changes in telomere length. Right? I think we'll get later into telomeres later, but basically we knew my PI knew Susan Bailey, which was one of the leads of the twin study experiment. And basically we just asked NASA, hey, we want to do something similar to what the twin study did, but with plants, looking at their telomeres. And eventually, after getting in contact with the right people, with Richard Barker, with Ryan Scott, through NASA Gene Lab, we found a team of researchers that were sending a mission up to the ISS in very short time. So Dr. Sarah Wyatt and Dr. Imara Pereira took us on their team, and we sent plants to space with them, and we measure the telomeres of those plants. We got a very nice study out of it. And then after that, we were asked, okay, what next? We had more questions than answers after this project. So we wrote another proposal. It got funded, and the experiment actually just came back down from the International Space Station. So that is how I became interested in the field of space biology, through basically having the right connections, knowing the people that are doing these projects, and being very lucky that this field of space plant biology is very collaborative.
Well, that's a wonderful story. So basically, it was a chance and that you got into that field and you are enjoying it, I believe.
Yes,
Still?
and now after, once you, it's almost, once you see those plants go into space and then you get them back in the lab and you look at the plants, you see them, it's like this is almost like my babies that they have been to space, right? And that feeling, like gets you hooked, right? You want to keep sending experiments, sending the next iteration. And I think it's a very critical item, especially for interstellar travel, that we need to keep exploring. And I have been lucky that after all of those experiments, and again, due to the field being very collaborative, I've been able to be a bigger part of it through the Moon Village Association and through NASA Gene Lab, other programs such as NASA Sign, NASA Star, BioAstra. So, on my free time, I do space biology research.
Well, definitely this is fascinating what you're talking about, but I have a simple question. It seems obvious to you, but can plants even grow in space or another planet?
So we have been growing plants in low Earth orbit for a long period of time. And plants do grow, and for the majority of the cases, they grow well. They are a tiny bit stressed. However, the plants are able to grow, they're able to generate fruit. And in low Earth orbit, they're able to do their function, which is, they can potentially generate, be used as a food source. It's not the major food source being used by astronauts, but astronauts have been able to eat some of the experiments and plants that have been sent to International Space Station. They as well are able to sequester some of that carbon. They're able to generate oxygen. And I think the most important one, which after you talk with astronauts in the International Space Station, is that they are a mental health support system, right? When you talk with astronauts, or you see their interviews, they always mention that their plant experiments is their favorite ones, the experiments that they're the most excited about to perform. And that is, after being enclosed on the International Space Station, which is it's a closed environment with your cram is tiny. Would love to go, but it's like it's a space in which nothing reminds you of home, right? And you see something green, and it reminds you of your backyard. So that is a very powerful use of plants that we need to keep into very special account now, especially that we're planning for missions that are over 1 year. And mission to Mars proposed around 3 year round trip. So for those trips, we're going to need plants for many uses, and one of them is going to be mental health support.
Wonderful. So you mentioned about, you said low Earth orbit. Care to explain to the listeners and audiences that what is a low Earth orbit?
So the low Earth orbit, it is basically the area in which the International Space Station is located, right? It is a specific distance in which the International Space Station orbits at around 17,000 miles per hour, and what holds on that specific area many different satellites and many different as well spacecrafts, so to speak. The low Earth orbit is the area in which as well we're still protected from radiation. There are different, we still have like a I believe it's magnetosphere that is protecting us from those space radiation damaging effects. However, it is what we have been studying, but I believe we need to go beyond low Earth orbit, move away from that protection that the atmosphere provides us, and go beyond low Earth orbit and start seeing the real effects of space, which will come with higher damage from those space radiation, those GCRs, those higher charged particles, right? So everything that we have studied in space has been in low Earth orbit, and thanks to marvels of engineering like the International Space Station, like the Mir, like the Salyut, like the similar to the Chinese space station Tiangong, low Earth orbit, we're still somewhat protected from the damaging effects of space on that low Earth orbit, but now we need to go beyond it. We need to go to the next frontier, and that is moving away from it.
Okay, so do you use soil and water to grow plants up there?
So
How do
So
you grow
actually,
plants?
on our experiments, we use what is called the VEGGI hardware. And for our experiments, we use Arabidopsis thaliana. And Arabidopsis thaliana, you know, there's always the joke that the natural environment of Arabidopsis thaliana is on a MS Petri plate, right? Which is like a media that has.
So it's a model plant that botanists use for studying different
Exactly.
kind of
It is
things, right?
the mice of plants, so
Yeah.
to speak. It is the model plant species. It is a plant. Yep.
I like that
It's a
analogy.
plant that
Yeah.
grows well, that the
Yeah.
genome is fully annotated. There is a big library of mutants available for purchase. And on top of that, have that these plants are very similar to other model crop plants, such as, hold on one second, I have the dogs fighting. Oye, get up. 1 second, I'll be back.
Well, now our video should go viral.
1 second, I'll be back.
Okay.
Fighting. All right, sorry about that. Yeah.
No problem. I will use it to make the video viral. Yeah.
Hey, use it. They're able to open the doors of my office somehow. So there you go. Yeah, intelligent dogs. Sorry, what was the question again?
Yeah. So we were talking about that. Are you using soil and water like we do it on Earth? Yeah. How do we grow plants in space?
Our experiments that we have grown plants on the International Space Station, we use the VEGGI hardware, which is one of the platforms to grow plants. Sorry about that. 1 second. One more again.
No problem. Take your time.
When the mailman comes, madness happens in the house.
No worries. No worries. I will use everything.
Yeah, make it go viral. Make my dogs be the meme of the internet.
Yeah, so I'm waiting for that break. So thank you for giving me one.
So our experiments, we have grown plants on the International Space Station. We use the VEGGI hardware, which is one of the platforms that is being used to grow plants on the International Space Station. It is very similar to any growth chamber that we can find here on Earth, but it is adequate for the International Space Station. So our experiments, we grew plants, Arabidopsis thaliana seeds on MS plates, which a lot of people think that Arabidopsis thaliana natural habitat is this Petri dishes with MS media on it. So basically these plants grow in this media that contains everything that they need to grow, right? But there are many different hardwares on the International Space Station that they use as well. Different media, different sources to grow plants on space. So the normal soil that we use, like potting mix here on Earth, it can be used, but it needs to be engineered to make sure that the soil doesn't go all over the place, right? Because otherwise, you're going to get the International Space Station very messy and full of soil, and we don't want that, right? But yes,
well, I don't think it's already very clean either.
Yeah, there's like a lot of bacteria around, right? On that new cell paper by Katie Rubins. Yeah, and there's like hydroponic as well system that has been developed, such as ponds, and there's like the APH, Advanced Plants Habitat as well, that it is the next iteration of plant growth hardware. There is hardware that as well is a fluorescent microscope to measure plant growth and different signals that the plants emit throughout their life cycles. The International Space Station has different hardwares to grow plants, and almost every sort of experiment has been adapted. So right now, actually, on Crew 11, I have a couple of friends and colleagues that they are sending wheat that is growing on Martian regolith simulant on the International Space Station, and all of this mission is sponsored and prepared by Starbase Brewing. Basically, they want to be the first beer on Mars, and they're doing this experiment to see basically how, what is the profile of that wheat and of that soil, right? To make something as fun as beer, right? So there is many different experiments going on, many different ways to grow plants in space, and in which plant scientists and engineers come hand in hand to make sure they grow adequate for the different missions.
All right. So you did mention about that. This is my second question, but you already touched upon that. So you talked about this platform where the plants are being grown in International Space Station. Can you describe a bit about, you talk about the VEGGI platform, or is there any
so
other place?
VEGGI is the one that
I
am the most knowledgeable of, because both of our experiments use the VEGGI hardware. So the VEGGI hardware, what people don't know is that, most people don't know is that whenever you do an experiment on International Space Station, you have a VEGGI hardware that is on the International Space Station, and you have basically its twin brother, which is in Kennedy Space Center, right? And whatever conditions is on the VEGGI on the International Space Station, it has a monitor in which that monitor will tell that VEGGI on the ground, which will be our ground control, how it has to behave, right? So carbon dioxide levels on the International Space Station changes, right? So that sensor will tell the sensor on the ground on the VEGGI hardware to basically adjust it, to basically meet the similar conditions as its space VEGGI brother, so to speak. So with VEGGI, we are very lucky that we have wonderful people that were on our team, such as Jeff Richards and JC, in which they have been working in VEGGI for a long time. And VEGGI allows you to really modulate the light intensity and the light source that the plants receive. Very similar to most of the growth chambers, but obviously putting things in space makes everything much more complex. And then once the plates with the MS media goes into space, you get into a very exciting Zoom call, which is the astronauts putting the plants into the VEGGI hardware. And after the experiment is finished, the astronauts will pick up those Arabidopsis seedlings and then freeze them so we can study them back here on Earth. Previous to that, there has been many, we have been growing plants in space since the 70s, right? In many different fashions, shapes, and forms. So one of the very early hardwares that was used in the International Space Station, it was the brick hardware, which was, so to speak, a brick, a canister, in which you have many different Petri plates that they could fit, and then they put eventually an LED onto it to give it some light. But then every year, and with the collaboration of many different industries, there is different hardware that is being developed to grow plants, developed by Redwire, by Sierra Space, NASA itself. So, I think every new iteration to grow plants in space is very important.
I'll keep saying names, all the commercial companies, so I can claim afterward.
Exactly, tag them on it.
Yeah, that's what I'm doing. Okay, so you worked with the Arabidopsis. So can you give us some taste that what kind of plants have been grown in under space conditions, that people can relate to? Is there any plant that Arabidopsis is a very special plant? It's your baby, but someone that can relate to that. A common man know that this plant has been grown in space.
So Arabidopsis, as we mentioned, it is. In my opinion, it is a very important plant for studies, but it is probably the most boring plant for it, because you could eat it, but it probably doesn't taste great.
Yeah. I didn't say that. It's Borja who's saying that. Everyone listening is Borja. No, I don't say Arabidopsis is boring.
I can say that because I've been working on it for 10 years. No, but still, I wouldn't have been able to get my experiment done if it wasn't for Arabidopsis. But it is, it is a plant that its family, the Brassica family, has very exciting plants such as broccoli, cauliflower, so very, very good vegetables that are very good for my daughter to eat, instead of Arabidopsis. But yeah, so there has been many experiments in International Space Station and other Salyut Space Station and Mir that they use different crop plants. So obviously, Massa and all the team that are growing different varieties of lettuce with starting with in 2015, I think VEGGI 1. They grow red romaine, they have grown green romaine, dragon lettuce. There has been as well cabbage that was grown on the VEGGI, Mizuna as well, so Brassica rapa. We has been as well wasabi mustard greens as well, so Brassica jansen. There has been wheat as well, rice, potatoes. So potatoes, obviously, for Russians is extremely important for many different.
And for
for
tortilla as well.
exactly. Yeah, I would love to make tortilla. Be the first one to make tortilla in space. Might not be a great one, but yeah. As well, what else? Radishes have been growing
Yeah.
International Space Station as well. There has been um, soybeans as well, very important one, especially for calcium levels. And then there has been actually personal experiments by astronauts. Astronaut Don Petit, he was always very keen in bringing some plants with him. So he brought sunflower seeds, he brought broccoli, and I think he brought zucchini as well to the International Space Station throughout his different missions. I know as well on Apex 09, I believe, Brachypodium was as well used. So this will be like grasses. So I'm probably missing some other crops, but I think those are the most important ones. And there's like a beautiful group at NASA that is working in the what they call crop readiness review, right? Which one of these crops could be very important are ready for flight, and what could be, how will they behave in space?
All right. So I think people have a taste of it, like what plants they can relate to. So far, I think potatoes stand out in that list.
Yeah.
Okay. So now just give us some idea that what kind of changes occur physiologically and at the cellular level once the plant, you mentioned earlier that they are not so happy but they still grow, so what happened to them in space?
When plants go again to low Earth orbit, very important, right? We're going to the ISS. These plants are able to grow, they germinate overall very, very well, and I'm talking about regular wild type plants, right? Not mutations or anything like that. So they regularly germinate very well, very close to full percentage of germination. And I'm going to make a long list of assumptions based on Arabidopsis, but obviously a lot of these different plants follow suit on that sense. They grow well, but they suffer what is called from oxidative stress, which is basically these oxygen radicals on space due to microgravity, due to high carbon dioxide levels, due to elevate higher levels of space-relevant radiation. This oxygen that is in the plants can become, so to speak, radicalized and start damaging different components, molecular components. So plants grow on the International Space Station well. However, they displayed signs of stress, mostly oxidative stress, that can have some effect on the biology of the plants.
Okay, so in general, this stress can have an impact, but still plants do grow. So that's the message that they
plants are able to grow well in space. They do have signs of stress. However, there's nothing that some genetic engineering cannot take care of, at least at the low Earth orbit levels.
Yep. We'll get into that later in the show. Yeah. So now, directly coming to the point. So your paper about plant telomere and how it was impacted. It's a wonderful study. Everyone should read. I will put that in the show link. So the first question is that what is a telomere?
Telomeres are the very tip of your chromosomes. They are very important for protection of the whole genomic material. So you can think of telomeres, and they have been described greatly by the brilliant people on the field of telomere biology, as the aglets, which is the very tip of your shoelace. So that very tip of your shoelace, if you take it off, your whole shoelace starts to degrade, and you cannot use those shoelaces again, right? So that's very similar what the telomere does. It protects the rest of the chromosome from being recognized by the DNA damage machinery and basically lead to eventual genome degradation. So the telomeres are protecting the rest of the chromosomes. Telomeres obviously are extremely important because, they have been a lengthening of telomere is associated with cancer cells. So one of the reasons why cancer cells do not die is because they have long telomeres, so they avoid senescence. And, if you have very short telomere, if you have very short telomeres, is one of the reasons why you and me we age. It is associated with early senescence.
Are we aging? Thank you for reminding.
We're aging well. Maybe not me. Maybe my telomeres are a tiny bit shortened due to a lot of stress. But yes.
Yeah, but thank you for the
Yeah.
reminders.
So
Borja.
basically, this telomere length, it is very important to be maintained in a specific range. It's almost like a yin and yang. Too long, it can lead to cancer cells. It's a hallmark of cancer cells. And too short, we start seeing early senescence happening. So you want the telomeres to be just right. And again, as we mentioned, telomere lengthening has been observed in astronauts in space. Sam Bailey and people like Chris Mason, they have been working on telomere length, and you could see that the telomere length happening in astronauts was almost independent of their mission duration, meaning that the moment they almost got to space, there was a telomere lengthening phenotype happening in space, right? Probably not this is a sign that the genomic material it is undergoing some changes, some modifications that are probably not great. For our body. And then that was the question that we set ourselves to explore. Is this telomere length happening as well in plants, right? Because beyond humans, there had been another experiment that measured telomere length, and this was in C. elegans, so in worms, right? And they saw similar phenotype, that telomere lengthening happening in worms, telomere lengthening happens in astronauts aboard the International Space Station. So in plants, we actually were shocked that we did not observe any telomere length happening in space. And I think this speaks to the resilience of plants to molecular changes, right? As you know, plants cannot move. They have to deal with the different environmental stressors, on a different way, right? If you and me, we're warm or we're cold, we try to move away from that source of environmental stressors. Plants cannot. Plants have to stay. And what they do is they deploy a very smart molecular mechanisms to deal with all of these dangers, right? So plants are probably better adapted to deal with those different environmental stressors and keep the genome more adequate. So in this case, the fact that telomere length did not change in plants, I think it speaks to the resilience of these plants to get their genome attacked, so to speak, and change. So, and we have seen that plants did not change in telomere length in many different other environmental places. However, when we put these plants, I imagine we are going to go there in lunar regolith simulant, there was a complete different story. I don't know if you want to cue that in, but yeah.
Mm-hmm. I think we will keep it for the later. But basically, from your explanation, you did mention the twin study where Mark Kelly and Scott Kelly were selected. Which one was on the
I think
ground?
for this, both of them are astronauts. Both of them have been in space. Scott Kelly was the astronaut that went to space.
Yes. But
so Mark remained on Earth.
Yeah, exactly. Mm-hmm. Yeah. So the Mark Kelly was on Earth. So the lengthening of telomere was there, and you did mention the C. elegans were there. So what I'm trying to figure out that your understanding or what you're trying to tell me that plants, unlike animal cells, they behave differently because they are not locomoting from one part to another because they cannot avoid the stress, whatever they are being exposed to. So they got some resilience. If I agree with your hypothesis, what about those microalgae or photosynthetic organisms that can swim from one part to another? Do you think they will behave like more of animal cells, like their telomere?
That is a great question that I'm not sure if I am fitted to answer here. But definitely what I can attest is that, in places where you don't think plants could grow, plants are able to grow, right? And I think we're going to talk about that in a bit, especially, seeing that plants can grow on lunar regolith, like on genuine lunar regolith, right? Like Annalisa Paul, Robert J. Farrell. So it's like plants are amazing organisms from a molecular point of view, right? We have plants that are 6,000 years old, right? And are still growing, they're still generating fruits, right? So, what? I wish I could live maybe 6,000 years, but I know probably I will not be able to live for 6,000 years, but plants can, some of them, right? And that speaks to their molecular mechanisms, right? So they are amazing organisms.
Because I think the telomere also actually have a lot to do with the cell division. and how it is established and the initiation of senescence. Because those microalgae I'm referring to, I worked with Euglena gracilis, this photosynthetic flagellate. It doesn't really fit into that algal lineage that has been used for aging studies. So that's why it just got me curious that, that would be interesting what you're telling me. Okay, so basically you're finding...
No.
Yeah,
I was just going to mention there's an experiment developed by the SETI Institute by Dr. Agata Zupanska that she developed a mission called Artemis. So Artemis basically is this algae that has been able to grow on the Arctic, right? So sub-freezing temperatures. And on top of that, they were hammered with very, very high levels of radiation. Way higher than any tolerable source by most organisms. And then these plants were as well sent to space and see how they behave. So I agree with you. I think there's some algae and that like such as fish.
Because I think they are early plants because gravity vector remained constant, but the magnetic field took time to be that stable, and so they were exposed to higher levels of radiations. Perhaps that's the reason they put, for them it's like, okay, it's
That's
a memory from the past. They still have those genes.
We might talk about this. We just sent an experiment to International Spacestation in which we graph some of these exotic genes, right, that they are radioprotected and put them in plants, right? So, for example, tardigrades, which is another amazing organism, right, that they have been able to, so to speak, survive outside in the International Space Station, right? So basically, lessons learned from organisms such as algae, right, such as tardigrades, and learning what are those genes important to give resistance to all of these environments can lead to improvement in many other organisms, right? If we know what those genes are and how to manipulate them correctly.
Okay, yeah. So I think we are now coming back to your study. So basically what you showed in your study that you did not see any lengthening of the telomere. If you will go into this detail, which tissue was most affected, but the telomerase activity, the enzyme which is responsible for lengthening of telomere was overly expressed, but nothing happened. Care to explain what happened?
As you said correctly, we saw no changes in telomere length. However, the enzyme that is in charge of adding these telomeres at the end of the chromosomes, telomerase, was super upregulated, like its enzyme activity, right, to 150-fold. So, from a researcher point of view, especially working on telomeres for such a long time, it didn't really make sense. Why would you upregulate an enzyme that is very, very tightly regulated to 150-fold change? Like, there must be a reason why telomerase is getting super upregulated and is not leading to changes in telomere length. So what we observe is that if we modulate the activity of these telomeres to 150-fold, close to 150-fold change on the ground, these plants were protected from different genetic stressors. Right? So basically what the space told us is that plant telomerase, has potential roles beyond chromosome end maintenance and involved in protection from oxidative stress, which that was a very interesting finding for us.
Okay, so basically you did check the gene expression of that enzyme. Did you check the protein expression?
We checked its enzymatic activity. So basically what we did, we obtained the plants that were flown to the International Space Station, and we performed like a total protein extraction and we enriched for telomerase, right? So we found some buffer conditions that is able to enrich telomerase greatly. And then we just saw how active the enzyme is. So we did that with, it's called a trap assay that was in telomere biology is very, very common. And basically that assay explains how that protein extra that has telomerase enriched on it is able to do its job, which will be basically to add these telomere repeats. And we saw, compared with the ground controls, right? These are the same seeds. One of them sent to space, one of them remain in Kennedy Space Center, right? If we give them the probe to add these telomeres, right? How good are they at adding telomeres? And this space-flown ones was tremendous, like 150-fold change in enzyme activity. It was very, very ready to to add telomeres, so to speak. But then we found out that is not what I was doing. There was a lot of telomeres, but what we did observe it is that telomeres was necessary to maintain the genome protected.
So basically, that's an interesting that the machinery used to produce that enzyme, but it did not perform its function. Do you think it's an economical approach?
It didn't
from
perform
the plant?
What we think it was the main function, but it performed a different function. Plants will not upregulate something to 150-fold change if there were no reason. Plants are in some sense smarter than us molecularly. So it did upregulate at a post-transcriptional level, because we did some qPCR on TERT, on the reverse transcriptase unit of telomeres, and we actually didn't see a major change. So this is all at the protein level, right? Something is happening to telomeres at the protein level in space that is making it super active, and not towards telomere elongation, but towards genome protection. It was a very interesting and cool finding, and I think we have been very grateful by NASA has been promoting this paper in Nature Communications as what it was, which is space can lead to the uncovering of these moonlighting functions in proteins, right? Because this is such a, plants don't grow, they never grew in space, right? Unless we make them grow in space. And, plants have to adapt their molecular mechanisms to be able to live in these conditions. So that's one of the mechanisms that plants do in space. They upregulate their telomerase activity to protect their genome. What will happen once we go beyond low Earth orbit? That is one of the questions that we're trying to answer next.
My cosmic comrades, we will continue our intergalactic conversation in the next episode. Until then, stay curious and stay connected, as we are just beginning our odyssey into the heart of the universe.